Hello everyone, David Wilson here from Dr. Stem Cells, and in today's video we are going to look at another exciting paper. We're going to try and be brief again, and this paper is reviewing the difference between the benefits and drawbacks of stem cell therapy based on where the cells come from, so their origin or manipulation process. And they're also specifically wanting to look at immunogenicity. That means how likely are stem cells to cause an activation of your immune system when you receive them. Mesenchymal stem cells are effective in treating the diseases. We know this, and if you've seen some of my earlier videos, you'll be aware that we think like this. And we have the difference between autologous, which are from your body for your body, but then we have to look at HLA-matched allogenic mesenchymal stem cells. So they're stem cells from someone else, which you're getting. So you're really getting a transplant of someone else's stem cells. And in order to try and reduce reactions or reduce immune system problems from the one cell to the other, we can do HLA matching. So that means that we take the cell, we look at it almost in the same way that you look at blood whenever you do blood typing and matching, and you match the type of HLA expressed in the cell to the receptor on the receiving patient's body, so that they'll have a less chance of rejection. This being the case, then we can use HLA matched allogenic, so from someone else, mesenchymal stem cells, and they can aid in the engraftment of human allogenic kidneys with or without low doses then of an immune suppressant tacrolimus respectively. However, these HLA alloantigens are problematic because cell therapy uses more HLA mismatched cells than autologous for convenience and standardization. Now, this makes sense. If you know what autologous means, in other words, from your body for your body, you will understand that it is extremely unlikely for these cells to be able to compete with cells from your body because they have an exact match. There is no difference. Your cells are your cells and your body will not reject them. So they go on to explain why this is here. They say that in particular, these HLA mismatched cells are going to show increased antigen-specific T and B cells. So cells that come and are actually out on the hunt, on the prowl to try and destroy these cells that are known as being foreign. And they're going to have, therefore, reduced function, reduced viability compared to the ones that were matched. They can use CRISPR or Cas9 based, that's gene editing, to try and edit the cells. And then you can induce T cell activation in the immune system. Interestingly, despite this immunogenicity being limited to the cells with foreign antigens, the acclimation of HLA and allo-antigen sensitized cells may lead to an allograft rejection. So if you have been given these stem cells and you actually build up enough of this immune irritation or immune system recognition that it's foreign tissue, that can actually make you more likely to reject the kidney as well, not just the stem cells that are being given to try and help ease this process. So the summary here is probably common sense, but it's nice to see that science does actually follow what we would say is common sense. And we can see here that if you've better HLA matching or depletion of the HLA, so you remove the HLA before you treat the patient, you're going to find that the cell therapy can actually reduce repetitive transplantation. So you've less need for new transplantation of organs through the long-term survival of this cell therapy. This can be really important then for patients who are seeking allogenic transplantation, that is to say they're seeking a kidney from a donor. And this would usually be in the case that their kidneys have failed or they maybe need donor cells. So what is the takeaway from this paper? This paper tells us then that, as we would expect, if your body has more foreign material injected and is aware of it, that is to say it's HLA mismatched, you're much more likely to cause a problem with rejection in general. On the other hand, if you have an HLA matched, or even better, a gene-edited cell which removes the HLA irritation completely, your B and T immune system cells are not likely to attack, and that's going to give you a longer period before your body would even recognize the foreign tissue, in this case kidney, and before you'd have a rejection or a reaction. As we've discussed already in another video, patients receiving autologous stem cells may be able to push away the need for a transplantation by actually improving their own kidney function. So it's an interesting idea to combine these two different papers. There's a different paper we've discussed in another video where we see that intravenous transplantation of your own stem cells, so a stem cell treatment with a systemic treatment then afterwards. So we do the harvest in the normal way, we centrifuge it, we wash the cells, and we inject them into the veins. This may actually cause an improvement in your kidney function, hopefully cause you to lengthen the time then that you would go before you would ever need a transplant from a donor. Obviously, there's no guarantee, and in some cases these things happen either before the patient is able to get an autologous transplant or before there's any awareness that there's actually a huge problem. Some people present with end-stage renal failure, really, because maybe they don't go to the doctor often or because other reasons have held them back. I hope this was interesting for you. I find these very exciting topics. Join me again in the next video where we'll try and look at more to do with the brand new cutting-edge research on stem cells, especially mesenchymal stem cells, allogenic versus autologous, and we're going to continue looking at this in the next video. See you then.